I'm very glad that today we listened first time and seeing so many faces from Kerala, which we have not seen, but we are just working either on an email or WhatsApp messages. So first, I congratulate all this public health system of Kerala, from which start from health minister and goes down to very lower level of public health workers, healthcare workers. And each outbreak during the pandemic, we see a wonderful response from everyone, which is really kind of, I can say that we feel very happy to work with everyone here, because everybody take their work very, very seriously. And because of that, every time when any case is declared for every virus is detected first time in Kerala, and everybody say that why Kerala always. But we say that Kerala is alert, Kerala is active. That is why they detect the first case always. And I think that shows their very active response in each time, either is pandemic or in the monkeypox cases. So as Dr. Indu has said, this is a image of a BSL-4 facility in the corner, where our team work on any emerging infectious disease, and we keep working on preparing those risks or pathogens which can harm our country and then can arrive or emerge due to zoonotic or any other mode. In the middle, you can see two females working in BSL-4 facility. And as I see in Kerala, there is a very female dominating team. The same is here in NIB, and we have a huge number of female scientists and technical staff who work with a very dedication, and we deliver whenever it is required for any such kind of emergency. So whenever we talk about any new disease, and WHO says disease X, there is always a uncertainty, severity, and mortality, and that create a lot of stigma or fear and panic. And if you look at the priority disease of WHO, which talk about many diseases, and out of those 4 diseases are present in India, and COVID, CCHF, and also Nipah and Zika. And there are many which are in pipeline to have entry anytime to the country, and that's why we need preparedness all the time. And the questions come why the diseases are emerging, and there are a lot of reasons. And some of the reasons which we all know, that travel, urbanizations, poor infrastructure, deforestations, lack of surveillance, early warning. At the same time, we are improving on the testing system and surveillance system. So I think these are some of the reasons, and why preparedness is all the time very important, and which Kerala show every time they're better than anyone else, that as fast as we detect, we contain and mitigate these risks, we can control the disease and save a huge lives. And that is what actually mattered in the public health system. So before I talk about the monkeypox, I would like to give a small history about how the virus started. So we all know, we all has heard about the smallpox virus, and the structure we can see. This is one of the largest virus, and acute contagious caused in the past and caused millions of deaths. And it was believed that it existed almost 3,000 years. And there is a lot of studies was done to understand when this disease must have started. And based on the literature, it seemed that it started in China in 4th century, and in Asia in 7th century, and in Minor in 10th century. And those people who got infected, 30% of the people died, and people suffered and had their scars even if they survived. After long efforts of decades, WHO, World Health Assembly, declared the world free of this disease in May 1980. And this took a lot of efforts to go through. So if you look at the all pox viruses, this is a representation. In the beginning, you can see the monkeypox in the red, and then Vaccinia virus, the virus which is being used for the smallpox vaccine. Then we are here in the green variola virus, which is also called smallpox. And then we can see camel and many other viruses, including cowpox. And then we have some other pox viruses found in rats and rodents. So if we look at the phylogenetic tree, which was built in the past, the sequences from smallpox, which was done from India, fall into P1 genotype, and there are 2 genotypes of the smallpox viruses, and they go hand to hand. And then there are number of the pox viruses, which is in the tail, which you can see here, that also include monkeypox is one of them. So the transmission of smallpox was considered to be droplets, oral, laser, pharyngeal mucosa, transmitted through a prolonged contact with infected patients, fluid or contaminated object. And in temperate areas, the cases were high in number, and India, there was a time India actually has 60% cases of the world. If you look at the virus, how it has reached, so we know the history, how people try to capture other countries and they reach to other countries by traveling. And in from 6th century, from Japan, then 7th centuries to reach to Africa, Spain, and then further it spread in Europe in 11th century. In 15th century, even Africa and the rest of the part of Europe and Africa was infected in 16th century. In 17th century, it reached to North American, and then finally it reached to Australia. 50% of Native Americans died because of the small infections. In India, if we look at this map, which was drawn around 3241, you can see that a large portion of the states were highly infected with this disease. And then after the freedom of country, we had many diseases which were affecting the health of the country, and smallpox was one of them. Government of India launched a national smallpox eradication program with a focus of vaccination, but it was not very effective. The cases were still very high. And in 1970, there was a liaison done with the government of India and World Health Organization, and a team of the experts came from foreign countries, and they worked with the Indian government officers and a huge team. They went for the searching, finding the cases, contact tracing. It is a beautiful story of one of the deadliest disease to get rid of from the planet. These are some of the posters which was made in that time, how to differentiate between smallpox, chickenpox. Because of the chickenpox infection, people were a lot hesitant to take the smallpox vaccination, and a lot of efforts which was done by, again, the public health worker contributed that we become free of this virus in 1975. So this is one of the very historical picture with the last case of the smallpox in our country. And we got this certificate of the zero pox during this period. So if we look at this map, which describe the presence of smallpox in different countries, initially, I don't think India didn't have the cases, but there was no method to report. So they are not recorded. But if you look at in 1926, the India is completely dark red, and that shows a very high prevalence of the disease that time. But at the same time, when the vaccination was implemented, we were totally green in 77. So how we got it out of this, and there was a series of the vaccine which was made, and that has again a beautiful story, that how Jenner made the first vaccine using the cowpox virus, and it started from passaging one human to another human, then from animal, and then it moved to the cell culture, and different kind of generation of the vaccine was made for the smallpox. And now currently third generation of the vaccine, which is being used in USA, is called a Bavarian Nordic. It is the manufacturing and also the KM Biologics, and these are actually available for the certain countries, but it is not available for the India. But at the same time, these vaccines are approved for the adults, and especially for Imavanex. This is a 2-dose, 4 weeks apart vaccination, and can be used for pregnancy, lactation, immunocompromised individuals. The next vaccine, which was developed by Japan, it is not a kind of replicating competent virus, but it is safe and even can be given to infants and children. And the method is bifurcated needles, the old one, which was used in the past. There was another vaccine is called ACAM20, which is a very popular vaccine, was developed. It is approved for adult populations. And it is, in the past, it was also produced by Sanofi Pasteur and produced in France. Then another first generation vaccine was basically made in the animals, and then it moved to the but the initially the vaccine, it has a lot of side effects. And the second, it was having a lot of containment because there was a mix of different strains, and it caused a lot of issues with the health. But still, it was better to save life rather people survive. As smallpox is considered one of the highest risk group pathogen, and a very favorite choice for the bio warfare. So many countries kept stockpiling these vaccines for a very long time, and they had a huge stock before this outbreak started. Currently, US is using these two vaccines of third generation and second generation, and it is available with them. But Korea and other countries also have the access of these vaccines. Just to clarify, India don't. So this is a picture of global smallpox eradication in the 75. The last case was reported, imported case from Bangladesh in India, that was in 75. And later on, we didn't have any cases in our country. So after this virus, then another member of this family is Vaccinia virus, which was used for making the vaccine. And it is still being used in different laboratory to use as a vector for making different new vaccines. And recently, the Vaccinia virus, Ankara strain attenuated virus is being used for making third generation smallpox vaccine. There is one more virus which is very relevant to our population. It's called buffalo pox, which affect buffaloes. And again, it is a zoonotic disease and transmitted to human beings who come in a very close contact of these animals. Not only it was detected in 1934 in India, but it is also present in Bangladesh, Indonesia, Pakistan, Egypt, Russia, and Italy. And it can be spread by sandfly and visit. But the origin of smallpox and how it must have originated, no one knows, and there was no record available. People even try to look at the mummies and other smallpox infected specimen, which was kept in different museums to sequence, but it's still there's history is still a history, and no information you know. So if you look at the buffalo pox distribution and prevalence, India is one of the country where buffalo pox is prominently present. If you look at this map, then different outbreaks has been reported in country in 85, 87, 92, 96. Even Maharashtra is one of the place where many outbreaks has been reported, and we have been part of many outbreak investigation. In 2008 and 19, in 9, Kolhapur district had recorded cases. In 2018, we further have detected the cases, and even we did the virus isolation from human cases. 6.6% attack rate was even seen, and also in the buffaloes where high positivity was seen. So these pox-like lesions were always associated with fever, malaise, and also pain at the site of lesion and axillary and lymph adenopathy is one of the characteristic of this disease. During this outbreak, we isolated the virus and we sequences the first pox genome from India, which was submitted and published. And actually, when we look at the molecular ancestry that we found, this is one of the very closest strain to the Lister virus of the pox, which is being used for the making vaccine in the past. So the pox viruses have a very close relationship. Even in the Vaccinia virus, there was a doubt if Vaccinia virus is good enough to protect the smallpox, because in the past there was no genomic sequencing tool. But in later on, when it was sequenced, it was found that it is one of the closest virus which matched with the smallpox, and that's why it gave so good protection. Even the vaccine gave 85% efficacy. So this is a sequence which we did in the past. And if you look at the confirmed monkeypox cases, so everybody thought that this is a disease restricted to only those 9 countries and 2 additional countries where the imported cases were reported in Africa, and it is called West Africa and Central Africa. But with the time, there was cases we were imported, there were animals who were imported, and they created hotspot in other countries than these 11 countries. But recently, everybody is knowing that the number of cases are actually increasing, and that has caused a lot of concern. In the first time in US, there was outbreak was noted, and then single imported cases were recorded in 2021, 22, and even other countries like Israel, Singapore, United Kingdom has reported these cases. So if you look at now the scenario, which is every minute is changing, it is present in more than 89 countries, and more than 32,000 cases has been reported. And even India, you can see present in blue. So as much as the dark the map, you can see the highest number of the cases. At the moment, United States of America is having highest cases than Spain, and then United Kingdom. So the cases are increasing. If you look at the genome of this monkeypox virus, which is again one of the very huge genome of 197 kb, and it has many open reading frames, and genome is quite big. So if I look at the genome of SARS coronavirus, we can fit almost 7 SARS coronavirus genome in one monkeypox genome. So the genome is very big. So if there is a mutation happening, it is very difficult to decide in a very short time that how these mutations are going to affect the transmissions or other factors to the host or the virus. So these are the challenges where everybody is now working. So if you look at the reservoir of infection based on the earlier published literature, mainly the dormouse, squirrels, rats, and monkeys were found positive with these viruses. The first case of monkeypox virus was detected in 1970, but the first monkeypox virus in identification was done in 1958 in a laboratory animal. So for India, because this is a new disease and we have not detected, so we categorize as a risk group 3 virus, and that's why when we are handling any pathogen or doing any study, it needs a lot of precautionary measures because the young population is not vaccinated and they do not have any antibodies for these protection against this virus. So if you look at the transmission, the primary infection is from animal to human. That is why it is called a zoonotic disease. And it can also transmit from mother to fetus, which we have seen in case of the smallpox. But also the bushmeat can cause this infection, and human to human transmission in a very close context, the transmission can happen. But in the past, there was no literature which can suggest that sexual transmission or one partner to another, the virus can transmit. But there was a few literature where it has been recorded that one person has two female partners and they have been infected. So, but recently when the cases started happening in Europe, there was a huge burst of the cases. And as per the recent case history, it seems that 91 to 97% of the cases are those who have the sex from men to men. And these are, I'm giving a figure of different countries who are reporting the cases. So if we talk about the mortality and severity with the different kind of the viruses, the Central African strain was always considered to be high mortality, high severity, and West African strain was considered to be lower mortality. So, so there are many factors why these emerging diseases are affecting human being, and that is we are the human being responsible, and that main reasons are deforestation, climate change, and one of the very strong factor is considered is smallpox vaccination cessation, and that's why as soon as this the eradication program got over, we saw this emergence of monkeypox disease. The people who work on the virology field, they always thought that monkeypox can emerge as one of the threat in the future, and actually that become a true now, which we are seeing here. So whenever such a new disease emerges, there is always challenge to develop faster diagnosis, quicker surveillance, finding out the natural history of pathogen, biologies, transmission, diagnosis, and many research questions which come, and we have seen a whole plethora of these exercises during the pandemic when COVID and its variant of concern made everybody's mind spin all over the world. At the moment, many countries like India do not have access to vaccines or do not have access to any antivirals. And there are a lot of now questions that how this disease is going to behave, and do we need vaccine, and so on. So these questions has to be answered, and it has to, lot of efforts need to be made to answer those questions. But whenever there is a new disease comes, we need to do a risk assessment. Either it is in a hospital setting or in a laboratory setting, that how we are going to handle the situation, what kind of PP is required, what vaccination we can implement, what treatment protocols we can do. And similar, we always find at the same time, it is an emerging high-risk group pathogen. So it has to be being part of ICR 2005. We need to report the cases to the WHO through Ministry of Health and NCDC. And that's what we keep doing. If we talk about the diagnosis for the pox viruses, the real-time PCR method, because it is a DNA virus, so a real-time PCR method, sequencing, virus isolation, and electron microscopy is also one of the very standard tools but it is not very fast and around and quick. And during the pandemic, the good thing is that the country has developed a very great capacity on the infrastructure for the real-time PCR. We had more than 3,000 laboratory to do the daily testing for the COVID. So in that aspect, we are quite well versed. But the challenge was to have these tests in the place, because pox pathogens are considered to be high risk, and nobody share positive controls or any testing methods so that you can do it. So it was challenge for getting a positive controller to validate the test. And finally, after a lot of efforts, we could get those positive control. We developed 3 real-time PCR tests. One, non-variolar orthopox, so which can detect buffalo pox, monkeypox, or any other pox except smallpox. Then the next was to detect monkeypox, and then third, to differentiate between Central African and West African. So these 3 tests were developed. And then first we did a bulk stock piling of primers, probes, and positive controls. And then we started training our BRD laboratory network, which is a part of DHR and ICMR. And initially we trained 19 laboratory across the country, and including NIV. So now we have a 20 laboratory functional, including NCDC laboratory. And yesterday we have trained 12 more laboratories, which will be functional if the cases increase our situation demand. Then there was a challenge of differential diagnosis. So we also augmented these facility to test varicella zoster virus, HSV 1 and 2, and then introvirus. I know you all must be seeing a lot of cases of pediatric HFMD. So it was required to even include in the panel. Then even the SEER member state around, they don't have any testing for the monkeypox viruses. So recently we have done training, and we also shared the reagent with them, and they have also initiated the testing. With all this preparedness, we could detect not only the cases which happened in Kerala, but total 9 cases were detected, 5 from Kerala and 4 from Delhi. And then for the genome sequencing of these samples were done, a virus isolation was done. And then also we are further working to understand more on this disease. So this is the laboratory network of 32 laboratory across the country, and which covered most of the geography of the country at the moment to cater. And out of these, because Kerala has the highest bulk of the cases load, so now there is a 5 functional laboratories are also situated in Kerala state, which can test any of the samples. So there was a guideline was released on 31st May by Government of India on the monkeypox, which included the definitions and everything. And a confirmed case should be considered, which has to be matching the case definition, and also the laboratory confirmed by either by PCR or sequencing. So that become a very must role of the laboratory to confirm. There is a lot of discussion on what kind of samples should be collected for the testing. So most of the guidelines actually say that we should collect the lesion, roof fluids, and base and crust. But now we are seeing a lot of change in pattern of the monkeypox, which was not in the past. And in that view, actually, we are also recommending to collect the NPS and OPS either dry or with the VTM, and blood collections and urine collection is also recommended if possible. With this case, we are all knowing that the cases can prolong from 2 weeks to 4 weeks. The recovery phase is longer in some cases, and that's how the differential diagnosis very much matter. The PP has been recommended the same as the COVID, and we are all very well versed with the personal protective equipment. The triple packaging and putting a category B biological substance UN 373 under 6.2 class is being given for those shipment of the samples. The training for collecting the specimens and also on these practices has been given to the team. This is the first case data where we represent the different positivity in different 3 tests which I described, and also we are taking the RNSP as a control to see that the samples is collected properly. If we fail to detect RNSP, it means we have to go back and collect the samples. In the first case, on 9 POD, we could see positivity in urine, blood, and all the samples. So it means in the beginning phase, when the cases are coming, they'll have positivity in each and every samples. These are the first two cases, pictures, which is already available online as a preprint, and also it is accepted in Journal of Infection. And I thank each one of from Kerala government, from the public health, from the hospital, who participated in the study, and they really did a great job. And that's how we could capture the data, and it is published, and which define the lesions and how they look like that. But when we talk about the differential diagnosis, many times the cases which come, the chickenpox cases are very well defined. A very good clinician can just identify by seeing them. So we always request everyone to look at the pictures of those lesions, and then go for deciding what to be done. Then there is a very clear-cut guideline for the rash and these kind of lesions for the monkeypox, chickenpox, and measles. But recently, the paper which is coming from many from in NAGM and also BGM, they have shown that even sometime people can come as a monkeypox cases without any lesion or these kind of very obvious rashes. At that time, we have to really very cautiously judge the cases and send for the diagnosis. The same time, we also need to look at the differential diagnosis of enteroviruses and varicella zoster virus. Sometimes the pictures look very, very horrible, and we can think that it is a monkeypox cases. So why I'm recommending to take a nasal swab or oropharyngeal swab? Because I would like to just quickly give the data of the first 4 cases from Kerala and Delhi. And the samples were collected at 9th and 14th day. And you can see in these 3 cases, everything was positive, including urine, serum, EDTA, if we are collecting early the samples. But if we are collecting specimen at the late phases, then still nasal swab and all lesion samples turn positive, but blood and urine may turn to be negative. So the post-onset day of disease and their symptom are really guiding factor when we are sampling such kind of cases. If we summarize all these cases together, which I will just take you there. So this is the case 1 follow-up, which we did on 9th day, 11th day, and 16th day. You can see that on 9th and 11th days, we can see a very high copy numbers present in all the samples, including blood and urine, and which also continue on 11th POD. But when we reach to 16th POD, the sample is only positive for nasal swab, blister, and roof and base of blister. So as soon as they improve, the viremia start reducing, and on a POD of 20, everything turned negative. But this will not be the same for each case. The host will behave differently. And the recent paper, Patel et al. Has shown that there should be, in the past, people have seen a huge number of lesions in those cases. But in this outbreak, which is happening across the globe, there are people who are presenting with the typical lesions, but there are number of the people who are coming with either just 1 lesions or 2 or 10 lesions. And even number of sites can be very, very low in many cases. So identify such cases would be really very, very important when they are coming to the clinic or coming to the hospitals. So if you look at the viral kinetics of these cases, which were recorded for first 5 cases from Kerala, we can see that the lesion samples are the one who are actively viruses present for very long time. And in the first case, we could see virus present up to 20th POD. In the second case, we could see more than 22nd POD. In the third, further, we could see more than 21 POD. But in this case, we could see the virus was present for very short time. And the case who died, actually, we could collect only NSNTS sample, and it was also having quite a good virus load. So so that's how the NSTS also become very important, not only for the monkeypox, but also we can use for the differential diagnosis if turned negative for the monkeypox. So for from different part of the country, the 184 cases has been referred, and most of the cases are referred by these 22 states, and the Kerala actively having the highest number of screening of the cases. and then Delhi, and then Maharashtra state. And these two states has reported the positive case. And if we look at the clinical picture, then we can see the very common. So this is a summary data of all 9 cases, where we can see the fever, fatigue, genital lesions, and lymph adenopathy is the one. All these are the 4 very prominent features which we see in most of the cases. But we are also seeing sore throat, oral lesions, and also lesion of face, scalp, pain in devolutions, and also the genital swelling, dysuria are the other factors which is present in these cases. So for these 9 cases, and one case which is again a lot of questions were there because the patient died with encephalitis, and not there was not lesions or those kind of typical symptoms which we look in the monkeypox cases. So the genome sequences of this virus has been always considered 2 clades, West African, Central African in the past, and they have been, even the severity, infectiousness was higher in Central African clade. But as soon as the virus outbreak happened in March, and the molecular ancestry told that the European state somehow started in March from Europe, and they spread in a different part of the world. And the phylogenetic analysis say that the exportation of monkeypox virus from Nigeria to several countries, which happened in 2018 and 19. So recently, the data which has come up around 500 plus sequences, which is being deposited in gene bank. Now the virus has 3 clade. The first clade represent the sequences from 70 to 2017 called Congo Basin, and the second clade belongs to West African clade, having sequences from 70 to 2017, and the clade 3 are having a mix clade different lineages of West Africa, and where there is evolving mutations we are seeing. Initially, the virus was having 5 sub-lineages, which was called as A.1, A.11, B.1, and then further 1A and A.11. So, but now we are further seeing the bifurcation of B.1 lineage, which started in Europe, and now we can see 5 more coming lineages in B1 itself. So the virus is definitely mutating, and people are noticing those changes. If I talk about the 5 cases from Kerala, which has been sequenced, we found all these imported cases from UAE is coming to the A.2 lineages, and the A.2 lineages don't have many sequences. It has only a few sequences from US, which was recorded in 21 and 22, and then we have these Indian sequences. And among this phylogenetic tree, which I just explained, clade 1, clade 2, clade 3, so we have these 5 new lineages which are actually presenting the new outbreak which is happening across the globe. So if you look at this next strain clade, and now there is a new lineages because of the mutation in the virus of B1 is present, and India is coming in A.2 lineage at the moment. So, so what is making everybody to talk about that? The DNA viruses generally actually cause 1 or 2 substitutions per genome per year, but we are seeing more than 46 SNPs now, and there are 24 non-synomies, 18 synomies, and these are the changes which actually making everyone think that how these mutations are going to change the susceptibility, transmissibility, or other factors of this virus. So from the first 2 cases, we could do the virus isolation, and we got the virus isolation in Vero cell. But we did the susceptibility study, and we could see the virus is susceptible in 8 different cell cultures, and that showed that how fast virus can grow in different cells. But at the same time, we also looked at this virus in electron microscopy, and you can see in the bottom this picture, which is a very typical size and shape of the pox viruses, which is done in an IV this is one of the very beautiful picture of the virus which is coming forward and looking very nice. So what is a way forward for dealing with future pandemic and outbreak? Definitely we have to alert, and we have to do the early identification. We have to work as a team to strengthen the One Health research, risk communications, strengthening capacity for source identification, quarantine, isolation, which we have shown and again that we are doing a great work the Kerala government, NCDC, IDSP, and ICMR, we all work together to find out whenever there is a requirement of any disease, then capacity building of laboratories for the rapid diagnosis, enhancing the production of the vaccine. India is a hub for vaccine manufacturing. We have to invest more and more in doing the research and R&D, making these a new vaccine for infectious disease, and we don't have to look forward to rest of the world that we borrow a vaccine and we are dependent on that, and we know that nobody will give when the emergency comes. So we have to really work in this direction, and also the sensitization of healthcare worker by different trainings which Kerala government is doing. I talked today Dr. Vidya, and she told that how wonderfully they are doing the training of ISC in their healthcare worker and rest of the public. So I think it's a great work by Kerala government. But at the same time, we have to work for the biomedical research, social and behavioral research, operational research. And in this direction, NIV is in the process to develop our own serological assay, because they except CDC, no one has any serological kit which can be used to trace the asymptomatic infection, or how long the protections will be there in these cases. Are those people who have been vaccinated in the past for the smallpox, what are the response they have? So we are in a process to develop ELISA, PRNT, and these assays will able to help not only those people has a past vaccination, but what is happening to the new cases, or what is happening to their close contact. What now WHO documents show that there are 11% asymptomatic infection also reported. So in India also we need to look at these how much asymptomatic infections are there with these cases. At the same time, there is a data which represent that once the cases are isolated and identified, the transmission become very low. But there is a new paper just recently came which show that once the person is infected, the kind of viral DNA is spread in the room, in the area where the healthcare district to them is actually humongous. So proper disinfectant practices and PP is very much required. With this, I would summarize my talk, thanking the Honorable Health Minister from Kerala, Madam Tinku Biswal Madam, for a great support during this outbreak, Dr. Asa Thomas Madam, then IDSP official, which I cannot name, and the DSO of Thiruvananthapuram, Kannur, Kollam. They did a wonderful work during this outbreak, and the team from GMC, Thiruvananthapuram, GMC, Kannur, and rest of the people who are working and putting a lot of hard work in any outbreak whenever it is coming. I also thank Dr. Priya Abraham, Director of NIV, our former Director General Balram Bhargava sir, Dr. Nivedita, and my team who is in this picture. This is a small team we have who work very hard during any outbreak. I also thank NCDC Chief, EMR Chief, IDSP Chief, and we have today Dr. Himanshu Chauhan with us to represent the team, and also many other people who I cannot name, but they really did a great job during this outbreak. Thank you very much for listening me, and I would be happy to answer if there is any question.